Okay, so for the evaluation of the explore phase, some of the positive components we noticed were that students are working directly with the cells and engaging in hands-on experiences using an important scientific tool, which is a microscope that they will likely be using later on as they're learning more about science, and that they understand that scientists are using those kinds of tools when they are trying to learn new things. The practice of gathering and evaluating information without direct teacher explanation is powerful, and that's happening as students are recording data from their observations. And it's important to note that they are not expected to use correct vocabulary or terminology at this point, which ties into the mathematical practice number 2, which is reason abstractly and quantitatively. They're reasoning with kind of abstract concepts for them right now because they're not aware of the different names of the organelles or their functions in the cell, because they're still just exploring at this point. Students are making connections between their past and present learning experiences. They're integrating those prior experiences with their new learning, kind of generating some questions, exploring questions that they have, and they're kind of confronting their concepts, maybe some misconceptions that they may realize are incorrect, or that will be brought up in discussion that can be guided to correct those misconceptions. The other connection to math is making sense of problems and persevering in solving them. They're just sketching and describing cells to begin identifying differences between plant and animal cells. As far as persevering and solving them, they're having to really look critically and talk with their group members about, well, what differences do you see? Maybe try looking at it this way, and really engaging in that problem-solving process. A couple of things we thought might help improve this phase would be having the teacher keep in mind some big concept questions that they're going to use to help encourage and focus student explorations. These wouldn't necessarily have to be posted, but definitely kind of in the back of the teacher's mind as they're circulating and guiding students in this exploration of cells. So those questions might be something like, how does changing the magnification affect what you're able to see? Because we know that students will look at the cells with magnification of times 4, and then again with the magnification of times 10. And that would tie into the cross-cutting concept of scale, proportion, and quantity. Another question that they might ask is, why is it important that you're precise in placing the cells on the slides? The directions in our lesson outline very specific on making sure that the cell samples are not folded or anything like that, and that they're careful in placing them so that they can see everything they're supposed to be able to see. And that question would tie into the mathematical practice of attending to precision. The last suggestion we had was in the lesson outline, there was no table or graphic organizer as there were in the other phases of this lesson, and that would help increase the usability for teachers and for students in organizing their sketches and notes, and ultimately allowing for greater clarity as students share their findings with the rest of the class leading into the explain phase.